Welcome to Unit 5, Genetics. Knowledge of blood group genetics is crucial for your understanding of certain types of reactions that take place in a patient, or when we're looking for donor units that need to lack certain antigens. So a sound foundation in basic genetics is important when discussing blood group antigens. Knowing the zygosity of certain blood group antigens can explain varying strengths of reactions when we do our in vitro testing. Knowing the pattern of inheritance of antigens can explain the cause of hemolytic disease of the fetus and newborn, and the chances of another fetus being affected in a future pregnancy. Some blood group antigens are suppressed, they're hard to detect due to position effects on different chromosomes. So in this section, we're going to provide you with a quick review of the basics of genetics and its application to blood groups. Blood group systems are groups of antigens on the red cell membrane that share related serologic properties and genetic patterns of inheritance. So we're going to be bringing back some basic high school genetics. A gene is the basic unit of inheritance. It holds the information that's necessary to build and maintain an organism's cells and pass genetic traits to offspring. So in this picture, you can see on the very left, there's the representation of a gene, a piece of genetic material. And then on the right-hand side, you see the chromosome where that information is stored. Chromosomes are those thread-like structures located inside the nucleus. Each chromosome is made up of protein and a single molecule of deoxyribonucleic acid, DNA. You have 46 pairs of chromosomes, 23 pairs of homologous chromosomes, of which 22 are autosomes. They're the numbered or non-sex chromosomes, and one sex chromosome, either X or Y, which will determine the gender. If you're XX, you're a female. If you're XY, you are a male. This illustrates the 23 pairs of chromosomes. Notice how they're numbered. For genetics, the term locus simply means the location of a gene on a chromosome. I like this picture just because it nicely illustrates we have an allele for purple, an allele for white, and what those bands represent are the locus or the location of those genes on a chromosome. You need to be very familiar with the terms homozygous or heterozygous and what they mean. An allele is one of two or more forms of a gene, and alleles have different DNA sequences. If you have a homozygous person, they inherit identical alleles. So these are some blood group antigens. AA simply means that the person is homozygous for group A. This is their genotype. The phenotype are the actual antigens we can detect. And again, BB would be homozygous, MM would be homozygous. Heterozygous, the alleles are different. For example, AO, you got an A allele from mom and an O allele from dad, and so on. So homozygous, they are identical alleles. Heterozygous, the alleles are different. Your textbook brought up dosage effect in this chapter. I usually like to save that for later on, but we're going to introduce it here and then we'll revisit it but later on when we're talking about antibody workups. Dosage is a variation in antigen expression due to the number of alleles present. In some blood group systems, persons homozygous for an allele have a double dose of the antigen on their cells compared with those who are heterozygous for an allele, and may get varying strengths of reactions with typing sera due to the varying number of antigens present. So remember, the alleles code for the antigens on the cells. Let's give an example. M and N are alleles. They're in a blood group system that we will learn about soon. Heterozygous individuals are M positive and N positive, meaning this individual would have 50% of their antigens being positive for M and 50% positive for N. Homozygous individuals have a double dose, meaning 100% of the antigens are the same. So when you're phenotyping an individual for M, you may get a 2+ reaction with heterozygous cells, M-pos, N-pos, and a 4+ with homozygous cells. They have a double dose of M due to the number of antigens present. Remember, in a heterozygous cell, you're going to have 50% of the antigens being one or the other. So when you add a drop of typing sera, because of the decreased number of antigen sites, you're going to get a weaker reaction. When the individual is homozygous and 100% of their antigens are of that one allele, then you would then expect there's more antigens for more antibodies to react with, so the reaction will get stronger. Some antibody specificities present in the patient may also show dosage, which is very important to note and explain whenever you're doing an antibody workup. So this is an example of antibody dosage. You will be getting into type and screen shortly. Screen is the procedure in which we screen our patient for unexpected antibodies. That's antibodies other than A or B. So we have all these antigens that patients may make antibodies to. These are the most common. So if you look at the green shaded in the middle under the MNS column, which is a blood group system, and then you go all the way to the right, you see that the antibody reaction with an M positive, N negative cell is 3+. That's a homozygous cell for M. It has a double dose of the M antigen. How do I know that? Because it's negative for N. In the second row, you see that there's a 1+ reaction. And if you look at the genotype of the cell, it's M positive, N positive, which means 50% is M, 50% is N. So again, when you have 100% of antigens on the cell being of the same antigen has a double dose, then you will many times get a much more stronger reaction with the patient's antibody than a heterozygous cell. Some blood group systems show dosage more than others. In talking about the inheritance of allele, a dominant trait is if the allele is present, the trait will be expressed. You don't need 2 of them, you only need 1. And if you have 2, it matters not, it's still going to be expressed. Recessive, you must have 2 copies, a double dose of the allele, one from mom, one from dad, for the trait to be expressed. The best example I can think of is sickle cell disease, and there's 2 alleles, big S and little s. If the patient is big S, little s, they are heterozygotes and they are a carrier, but they will not have the disease. So they are able to transmit the big S to a child. But if the other parent is normal, then the child will not get disease. In order for an individual to inherit sickle cell disease, they must inherit a big S from mom and a big S from dad. And that then they're homozygous and they will exhibit signs of the disease. So this gives you an example of traits. Traits are the observed expressions of genes. So you can see we have a carrier mom and a carrier dad, and the chances that their offspring will be affected because they have inherited 2 copies of that recessive gene. This slide is from the PowerPoints from the textbook, and I just like that it presents information in a different way, but the most important component of this slide is at the bottom. Genes that do not express a detectable product are considered amorphic. For example, the O gene. The O gene there's no such thing as an O antigen, but O indicates that there are no A genes, no B genes. So it's a big fat 0. And the same thing with the D typing. If you get a negative reaction with anti-D, we put a little d, and it's a placeholder. A little d just indicates there's no D antigen. There's no such thing as little d. It's an amorph. It just represents that there's not a detectable product on the cell. And sometimes that's a difficult concept for students to get. So here's a slide that illustrates the terminology we just used. Gene loci, the location. In this illustration, a large uppercase letter is a dominant allele. A lowercase letter is a recessive allele. And so on the very bottom, you can see which genes or traits are homozygous. Antithetical is the term used to describe allelic antigens, and they mean opposite. They're on opposite chromosomes. So Cal and Solano are blood group alleles. If an individual is heterozygous, there will be one copy of each allele on opposite chromosomes. Substitute K for the purple gene and little k for the white gene, and that would then illustrate the antithetical blood group antigens. So again, if the genes are on separate chromosomes, then the term used is antithetical. Polymorphism is the inheritance of genes or alleles in different forms. A gene is said to be polymorphic if more than one allele occupies that gene's locus within a population. In addition to having more than one allele at a specific locus, each allele must also occur in the population at a rate of at least 1% to be considered polymorphic. One example is the ABO blood group system. You're either going to have an A, a B, or nothing at a certain position. So that it's polymorphic because there's different genes that can sit in that spot. You're going to learn that the Rh blood group system is the most highly polymorphic blood group system we'll study because of the greater number of alleles. In classical genetics, synteny describes the physical co-localization of genetic loci on the same chromosome within an individual or species. Today, however, biologists usually refer to synteny as the conservation of blocks of order within two sets of chromosomes that are being compared with each other. Synthetic genes are defined as genes that lie on the same chromosome. Two genetic loci have been assigned to the same chromosome, but still may be separated by a large enough distance in map units that genetic linkage has not been demonstrated, so they can be independently inherited. Genetic linkage occurs when particular genetic loci or alleles for genes are inherited jointly, all the time. They're so close together on the same chromosome that they tend to stay together during meiosis and are thus genetically linked. For example, in the blood group system, the MNS blood group system, again, these alleles are so close together that the chances of them being inherited separately is near impossible. Crossing over is bizarre, and alleles at loci linked but located at some distance from each other will often be separated by crossing over. Crossing over occurs when two genes on the same chromosome combine and produce two new chromosomes. And the most bizarre mutation caused by crossing over is called cis-AB, where we are taught that the A and the B gene are inherited separately. But this mutation, which is very rare and usually found in certain countries in Asia, the AB genes are on one chromosome, so they're inherited together. So theoretically, you could have a group O mom give birth to an AB baby if the dad had a cis-AB. So we'll talk about cis and trans in a moment, but yeah, this can cause some really unexpected surprises. So genetic interaction, the location of inherited genes in cis or trans positions can affect the expression of the gene. Alleles on the same chromosome are cis to one another. Alleles on opposite chromosomes are in transposition to each other. So if you look at the first picture, it's trans. We have the D on one side and big C, little e on the other. In the cis position, the D and the C and the E are on the same side. And that's how I get these two separated, these terms. Cis, same side. Whereas trans is opposite side. So cis, same, cis, same. Anyway, that's what has helped me get these terms straight. As you recall from your high school biology, Mendel was, I believe, a monk who studied pea plants, and he did many genetic experiments to determine hereditary patterns. So he would cross-pollinate the different colors of plants, and he did the long ones and the short ones and all different colors, to where he really was able to do an excellent study of how traits are inherited. Mendel's law of independent segregation refers to the transmission of a trait in a predictable fashion from one generation to the next. Alleles at loci which are carried on different chromosomes, or at loci far apart on the same chromosome, and whose entry together into a sex cell is a matter of chance, are said to segregate independently. Another law is independent assortment, which is demonstrated by the fact that blood group antigens inherited on different chromosomes are expressed separately and discretely. So you see a mom and a dad on the top, and then their children. And you see the phenotypes. Again, these are the traits that are actually detectable, expressed, and the genotype, which is the actual genetic material that makes that trait. Phenotype and genotype are two terms that students frequently get mixed up. And I want you to know the difference between the two terms and what they mean. A phenotype is the assortment of antigens actually detectable on an individual's red cell. So I'm going to give an example where we take our anti-A, it agglutinates, that indicates the presence of the A antigen. The genotype is the actual genetic makeup of an individual, which cannot be determined with certainty except through family studies. So here's the example. A group A person has the phenotype A. The antigen is actually detectable with specific typing sera. Add a drop of anti-A, a drop of cells, spin, and agglutination indicates the presence of that antigen. The genotype, the actual genetic makeup, could be either AA or AO. This cannot be determined through the blood typing. The family would need to be studied to confirm the genotype. So if you had a mom who's group A and the dad is group O, then you would know the person's genotype would be AO. Let's look at two examples using the Punnett squares to determine genotypes. A group A and a group O have children. Remember, A and O are the phenotypes, what is actually expressed on the cells. Without knowing the parent's genotype, the genes actually inherited, you'll be unable to determine the children's genotype. So let's take the two examples. On the example on the left, a group A and a group O have all group A children. Then you know that the group A is homozygous for A. Otherwise, you would have a couple of group O's sprinkled in there. In the example on the right, you know that the group A parent is heterozygous in AO because half of the children are group O. And that would not be possible unless the group A parent was heterozygous for group A. I love blood group genetics. So let's look at an example of a pedigree, a family tree. The mother in the first generation has the AB gene since her phenotype is AB. In the mating for the second generation, the row below that, the genotype for the father could either be BB or BO since his father's phenotype is unknown. See the little question mark? So it would appear the mother may be homozygous A since both parents are A, but look at their children's blood types. What is the mother's genotype now since both children are B? She must be an AO. There's no other way to explain, unless the father wasn't the father, but we're not going there. We're going to pretend this truly is the father. So again, applying your blood group genetics to a pedigree, you're able to determine the genotypes from phenotypes. Blood group antigens are codominant. If the allele is inherited, it will be expressed. Zygosity does not matter. The ABO blood group system is a prime example of codominance. If you inherit an A from one parent and a B from another parent, they will both be expressed. So this is how blood group inheritance looks like in a Punnett square. So on the top we have a parent who is heterozygous. They're A and O. So you bring those down and write those letters in the rows below. On the left-hand side, we have the other parent being group O, and the only genotype for a group O person is OO. So you bring that across and write that next to the original letters. So you get 50% of the children will be group A, and 50% of the children will be group O. Here's another example, which is very interesting. This type of mating, you will get all the blood groups. So we have a heterozygous group A and a heterozygous group B. So 25% will be every blood type. 25% AB, B, A, and O. In parentage testing, codominant traits are very useful, and there's two types of exclusions in paternity testing. There's the direct exclusion and indirect exclusion. A direct exclusion is the case where there's a genetic marker present in the child, but it's absent from both the mother and the alleged father. So let's say the child is group A, the mother's group O, the father's group O. Where did that A gene come from? It must have come from the real father, as both parents are group O. Indirect exclusion is a little bit trickier to understand. This occurs when there are genetic markers that are absent from the child that should be transmitted by the alleged father. So let's say, for fun, the child is, their genotype is they're homozygous for FYA, Duffy A. The mother is homozygous for Duffy A. The father is homozygous for Duffy B. So what makes this an indirect exclusion is the child should have inherited the Duffy B from dad, but there's not any. Indirect, because the absence could be due to some rare Duffy genes that cause suppression of the expression of the Duffy antigen. If the dad is heterozygous and he's big FY, little FY, and the child inherits the little FY, it may make it appear that the child is homozygous for Duffy A. So again, there are some blood group systems where there are silent genes that could cause misinterpretation of a paternity test. That's why these need to be done with great care. Nowadays, we go ahead and do genetic testing, DNA testing, but the concept is still the same. There's direct and indirect exclusions. In parentage or paternity testing, a direct exclusion is much, much better than an indirect. Nowadays, DNA testing is now the gold standard. It's very cheap and relatively fast. And there used to be billboards everywhere, "Who's my daddy?" And you could meet together and get tested to determine the paternity of the child. Population genetics is a very important tool in our toolbox in the blood bank. To determine the genotype or phenotype occurrence within a population, two formulas are used. One is a phenotype calculation. This is the one that we use all the time in the blood bank. This enables us to find a unit of red cells with certain antigen characteristics, for example, antigen negative. The Hardy-Weinberg formula is a mathematical equation that can be used to calculate the probable genotype frequencies in a population and track changes from one generation to another. Punnett squares predict the probability of offspring genotypes based on the genotypes of the parents. The Hardy-Weinberg allowed geneticists to do the same thing for entire populations. The phenotype calculation is very important when you need to find compatible blood for a patient who has multiple antibodies. Phenotype frequency is performed by testing a population and determining the frequency of presence and absence of certain alleles. The phenotype frequency should be 100%. For example, if your patient has anti-JKA, 77% of the population will be JKA positive, 23% will be JKA negative. So you have a 23% chance of finding a compatible antigen negative unit of blood for a patient. So that's what, about 1 out of 5. The most common time that we use phenotype calculations is when patients have multiple antibodies against multiple antigens. Knowing the frequency of each antigen allows one to calculate the number of units you would need to screen to find antigen negative blood. This is performed by multiplying the percentages of each antigen negative allele. The phenotype calculation is most frequently used to determine the number of units you're going to need to screen to find blood that's negative for the antigens your patient has antibodies to. You must be able to perform this calculation on the next exam. You will be given the frequencies of the antigens involved. So in this case, we have a patient who has an anti-c, an anti-cal, and an anti-JKA. So the frequencies are 20% of the population are negative for little c, 91% are negative for cal, and 23% are negative for JKA. In order to determine how many units that you're going to need to test to find units that are negative is by multiplying the frequencies together. So you convert them to decimal form. So at 0.2 times 0.91 times 0.23, that equals 0.04. You then multiply that times 100. Don't forget this step. So you would have to test, if you were testing 100 units, you would find 4 units if you were lucky. Because unfortunately the red cells don't read the books, and these eventually you will get the number of units you need, but you may screen 100 and get 2, and then you screen 10 more and you get 4. The statistics will always work in your favor eventually. You may have to listen to this presentation a couple of times in order to get a sense of understanding of the material presented. But if you don't, be sure and write down your questions and bring those to class. Spend quality time with your textbook because it is well written and does a good job of covering genetics.